All right. It's a little early, but let's test the sound. How do things sound?
I can
Oh,
hear you.
oh, good. I can hear you. All right, so that's not broken yet.
Are there more joining, or do you not know of any more people that can join?
I put out another invitation. You're the only one who took me up on it, so
All right.
it, that's fine. I wouldn't want 50 people here, so yeah, we'll have a little conversation. I do have some stuff prepared that I'll go through that will hopefully spark some interest, but we'll see.
Sure.
Yeah. So let's see. Sound is okay. Looks like it's video's okay. I'm getting some complaints from YouTube about the bit rate. Don't know what's going on there. So I'm trying slowly to implement some of the many suggestions I got in our Sunday live stream about how to do this all better. So there have been some changes. So
Is, is this
less.
is this your screensaver? Because I can see like on the right, I see like folders and documents on the
Really?
screen. Yeah.
Oh, yeah. Okay, yeah. That is just my second screen. There's some stuff spilling over from my desktop there, but
All
no
right.
big deal. Yeah, yeah. And then in a moment I'll switch to putting up my little keynote presentation, and those will go away. I kind of like that it's keeping my little floating head up there. So we'll see if that holds up. It may not, it may disappear, but we'll do what we can. 5 more minutes. We'll give people time to show up. Who's having social anxiety? Alan Smithy says it's not surprisingly nerds having social anxiety around public speaking on a Zoom call. I have no anxiety at all about public speaking. I do have a little anxiety about the technology and making sure everything works. And yeah, usually when you do public speaking, you go to a place and they provide you a podium and a lectern and they've done all the wiring and they got everything set up for you, and you just get up there and you put on your microphone and you start talking. Here I have to play with gadgets. It's kind of tricky because I have to figure all this stuff out. So anyway, as I said, I'm implementing some of the suggestions. So I'm going to try and be less stodgy, less formal presentation sort of thing, and more conversations. So people should feel comfortable raising questions in the chat, and Nestle 20 can feel free to interrupt and ask questions and make his own contributions. So it's all very casual. I got rid of some of the background clutter, putting up a screen, so that's less distraction there. I am plugging things more. I haven't yet gone through and edited all the YouTube stuff. So people were suggesting lots of changes to, the stuff down below the video itself, and I'll experiment with that a little later. Let's see what else. Oh, and so what I'm aiming for here is to not get carried away. I could talk about this stuff for hours, but I won't. So I'm going to aim for 20 minutes to half an hour of just setting the stage, talking about these details, and then hopefully we'll get some feedback and get people joining in the conversation, which is always the fun part anyway. It is in my classes as well. This last year has been so disappointing because everything has gone to Zoom, and I haven't mastered Zoom yet. And so it's like my students just sit there passively listening to me as I
Yeah.
answer them.
I'm also having courses for my education program, and we also use we don't use Zoom, we use Teams,
Hmm.
and it's also a bit yeah, it works, but it's not as fun, of course, as being in the classroom with everybody
Right.
being right.
Yeah, it works, but there's a whole new culture I have to master to make it, you know, to bring out students. Usually when you're in a classroom, you've got a room full of students, and after a while you get to know them well enough that oh, this person will be a good person to ask a question of about this subject, and this one is pretty much guaranteed to chime in at some point, and you can get the riff going. I also do things in the classroom to try and draw them out a little bit. Like every semester, I have a couple of drawing days. I just come in with big sheets of newsprint and a collection of Sharpies, and I tell them to illustrate what we're talking about, make cartoons. Can't do that very easily, although I suppose I could. I could just say, okay, after we're done, everyone pull out a piece of paper and we'll see. Whatever inspires you. Okay, it is officially noon. So I guess I need to get to work here. So let's see what happens when I do this. Okay. Yeah, I got rid of some of the crap on the screen. That's all good. Is my face appearing on the screen? I don't know if it is or not anymore. Probably not.
I see your face on the live stream on YouTube.
Ah, okay. Good. All right. So anyway, here's the deal, that what we're going to talk about today is a fairly basic paper, this Protas and Patel paper from 2008. Um, it's kind of a general overview of the whole issue of color and patterns and these surface patterns that are so easy to study. And I'll also introduce a little bit from some other papers that I will also link down below when I'm done, that go into a little more detail on the mechanisms. So we're not going to get too deeply into the weeds today. We're just going to talk a little bit about pretty stuff. So, the Protas and Patel paper introduces all these great ideas that patterns and color are so obvious. They're ubiquitous in nature, and there's all kinds of cool stuff to look at. So we should be able to do that. And so the questions are like, why do zebra stripes look like this? Birds are spectacular at generating color in their feathers, and they're very important for things like mating displays and so forth. Some of my favorites are fish, tropical fish in particular. Wow, just this is remarkable stuff that they generate these spectacular colors. Uh, Mammals, I'm sorry, yeah, zebra stripes are kind of flamboyant, but otherwise, I'm not too impressed with mammals. We've just got a
And
couple of genes.
and
Yes,
that's probably one obvious reason why mammals are so boring, because they originally were colorblind.
yes, and many of them still are.
Yeah,
Yeah. So colorblind, the mammals went through a phase where they evolved as nocturnal animals primarily, we think. So they lost color vision, but they also lost any
yeah.
need for all this color. So we've
So,
got a
so,
few
so, yeah,
genes,
so the only patterning they could be useful is like camouflage. We often see like counter shading in
right?
mammals.
And
Of
disruptive
course, they are
camouflage.
yeah, they are the boring side
Yeah.
of patterning. Yeah,
Yeah, but for things like fish, on the other hand, they make great use of color. So
yeah.
cichlids, for instance, there was a study on cichlids, and I think they have 7 different color receptors in their eyes. So they are much more sensitive to nuance and shades of color than we could be with only 3 color receptors. But yeah, fish have been a subject of interest. So this is what was really an important issue for us in the zebrafish world. Back in '96, Development dedicated an entire issue to zebrafish development. I've got a copy of it sitting in my lab. It's got all kinds of juicy, great stuff. And they chose for the cover this illustration, which is a collection of zebrafish pigment mutations. So you might think zebrafish are actually they're kind of bland in their color, especially compared to things like cichlids. But zebrafish have horizontal stripes, but when you get close up to them and you look really carefully in the microscope, you notice there's patterns of iridescence in those dark stripes. They're not actually black, they're bluish black with little sparkly bits in there. They're spectacularly gorgeous. When you look at the pattern, especially when you look at things like the fins, look at all this variety. And yeah, and you get these golds. So zebrafish, for instance, have bellies that are either silver or gold, depending on your sex. And it took a while to get used to actually recognizing the subtle differences there. But fish, of course, are going to be finely tuned to it. So you got golds and blues and blacks and all these colors and these patterns. These are all mutants. So one of the things that got people excited about zebrafish is that we could generate all these mutants, and there were people doing research on pigment mutations. So you get these patterns in just their fins. And so the question is how? What's the basis for forming these patterns? Are there rules to how you can generate patterns in the fins? And people are still working on that. We don't have a definitive answer. Sorry. We kind of know what genes are involved. And as the Protas and Patel paper discusses briefly, there have been various complementation assays done in zebrafish to identify, which groups of genes are involved in generating these patterns. You can do cool things like cross these and ask what patterns you get. They can even do interspecies crosses. So the paper talks about looking at a different Danio species and being able to do the crosses with those and sort of do a complementation assays and figure out how many genes are involved in this process. But yeah, we got all these gorgeous fish. How do they? I mean, you can see some patterns here, some consistencies, but what's the underlying rules? But what I'm going to talk about mainly is let's just focus on butterflies, because butterflies are kind of awesome, right? And we actually know quite a bit about butterfly wings and how those patterns are. So everybody out there listening, just take a look at that figure. Can you see rules and patterns? Can you make hypotheses about what the underlying framework to these particular color patterns might be? People have been working on this for a century or so, just trying to discern, is there rules? Yes.
I have a long time ago I've seen some discussion about like the differences between pattern formation in butterflies compared to like leopards or something like that, like cats. And you
Yes.
see that the patterns, well, there is some regularity in the patterning of cats. There is still some like the left side is not identical to the right side. Like if you put in a like symmetrical, it's not perfectly symmetrical, but with butterflies, it's almost always perfectly symmetrical.
Yeah, and you can see
So it's
that in this
different
illustration.
rule. Yeah, there's a different rule going on in the pattern formation of butterflies. And
Yes.
I don't remember the name exactly of its pattern formation.
Yeah, but in mammals, you kind of expect that a lot of it is random variation. That, no two
Yeah,
zebras look exactly identical, just like no two
exactly.
fingerprints
Yeah.
look exactly identical. But when you look at butterflies, you can find consistent variance. let me emphasize the word variant, because you can go collect one species of butterfly and you can find lots of variations within that, but it's like variations on a theme. And so there's got to be some underlying structure, some informational matrix that's laid out across the wing that is just regulated in subtly different ways in different individuals. So this, for instance, is some work by Fred Neichhout. It's some really cool stuff here too. Uh, this is this is a review I could have chosen, and I thought about choosing, but it's so much more theoretical and so much more detailed. It was kind of overwhelming. Anyway, Fred Neichhout was doing these studies, and he did what I just suggested. He just, let's look at the butterflies, look at all the different versions, and see if there's commonalities. Is there a pattern to these that we can decipher? Can we find the underlying rule book just from on morphological grounds? And yeah, he says, okay, if you look in the nymphalid butterflies, that those are the most common form of butterfly out there. Things like fritillaries and so forth are nymphalids. And you look at them, you can actually see that there is this ground plan, he calls it, to the pattern. That, there's these three bands that go out like this, and there's the ocelli, the spots that aren't really eyes, but they're called eye spots, that are out there on the fringe. And different species and different variants within a species exaggerate or amplify or accentuate different portions of this pattern, but it's still the same fundamental pattern everywhere. You just switch on various things in various ways, or you turn off this set of spots and you turn on this set of spots, but the pattern is lying there in the wing all along, which is kind of a fascinating concept in itself. So Protas and Patel point out this lovely little species. This is, what was it? Kalina. Uh, they imitate dead leaves, and they do it pretty well. So once again, what Nightchat would say is that there's still have that same pattern, but they've dampened down the colors, they've switched some things off and some things on, but you could imagine they're building on the same structure. Now, also, as Protas and Patel point out, is you get this wonderful variation within a single species, which is kind of impressive. And they mentioned a functional reason for that might be, well, that's one way to make sure your predators don't single it, home in on one particular feature and use that to recognize you in your camouflage. I don't know if I entirely buy that. I kind of lean towards thinking there's more of a chance involvement here. Uh, Nightshot actually looks at that species and tries to interpret it. And yeah, and he puts it in the same framework as all those fritillaries and others, that yeah, this is the basic pattern. And what you're doing in Kalima is just accentuating different bits, but underneath it all, there's still this pattern. So for instance, that in the wing of Kalima, you get that what looks like a leaf vein running the length of it.
Mm-hmm.
And he's arguing that, yeah, what that is just this band that's being accentuated to form that vein-like structure.
Right. So
Yeah,
so it's like the components are already there, but they are just shifted in the right way to make it look like a vein.
exactly. So there are these underlying patterns, and that is the toolkit you have to work with as a butterfly. And you can choose to brighten this one up or dim this one down, or um, make this one more prominent and link up with other parts of the pattern. You can do all that kind of stuff, but you don't need to generate brand new features. And you get all that amazing diversity of butterfly shapes. Okay, this is another. Oh, this is Vernon French's work. Vernon French in 1997. Yeah. Uh, this is where we start getting into the mechanisms. How do you do this? And he was analyzing the different genes that are expressed in the wing. So remember, night shot is just here. Here's what it looks like. Here's my interpretation. Here's my hypothesis. Let's move on from here. And Protas and Patel are just describing the amazing variety of forms that you've got. French in 97 was trying to puzzle out what are the genes involved here. And
These are
this,
the imaginal disc of the
yes,
wings. Yes.
yes, it's a gene expressed in the imaginal disc. He noticed that, yeah, and it's also correlated with other important features like the expression of veins. So you got this venation pattern in the wing, and that's all interlinked with the pattern of scales to produce the spots and so forth. And so, for instance, he's identifying these things called focal spots, and you can see that there's patterns of scale expression making a little circle right there, and you could ask what exactly is at that dot. And his answer was it's a gene called distal-less.
Hmm.
I don't know, have you heard of distal-less before?
Yeah, I've heard of it.
Yeah,
It's of course the name sounds like when you mutate it, it deletes a distal end or something.
exactly. Yes.
Hmm.
Distal-less was first identified as a mutation in limbs of Drosophila. So this is just to illustrate that. So here's a Drosophila leg.
But before moving on, somebody in the live chat says, would it be fair to say that it works like an LCD screen? I think the patterning of the wing.
Oh, oh, that's interesting. It's a lot less finely grained. I mean, an LCD screen is just going to have this regular, crystalline pattern of the same 3 or 4 sensors or light emitting diodes all throughout. Uh, this is kind of on a cruder scale, but yeah, you got a pattern, an underlying pattern, and do you switch it on or do you switch it off? That's that's part of it. Okay, so distal-less. Distal-less is a really interesting gene. Yeah, it was. Yes, as Nestle 20 perceived, it affects the distal structure of the limbs. So if you mutate it, this stuff fails, and you get little stubby legs. And its expression can be traced back into the imaginal disc. So you all know in fruit flies, there are these little clusters of adult tissues that are preserved, maintained, generated in the larva. So it's got like 15 different imaginal discs. And at metamorphosis, what happens is those imaginal discs move to the outside and sort of evert to form the limbs. So this is a limb disc right here, and all this is cuticle in the larva, and it actually is a flat disc. And what it will do is it will actually pooch out in the middle and extend to form the leg or the wing or whatever. So distal-less, as you might expect, is expressed right here.
Hmm.
So if you don't have distal-less, you're messed up there. But yeah, this aversion is kind of important. So this is another example. This is the imaginal wing disc. And again, it's a flat disc. That's what they call discs. It's very flat. And this little portion is going to form the wing. And what it will do, if you look at it in section, is it's just kind of puckering out there. And then at metamorphosis, this just kind of springs out. It expands. Uh, I can see this routinely in my genetics class because we're raising flies. So one of the things you look for is right after pupation is complete, they emerge, and their wings are these rumpled little balloons. They look like little saggy balloons hanging off their chest. And over the course of a few hours, they inflate them, and then they flatten to form the wing blade. And they have a dorsal and a ventral side, which have different molecular markers. So that's
Hmm.
also kind of neat.
Also, the same person says like a telescope, like
Yes.
when the black pulls out, it's like a telescope when you pull out the layers out like that.
Yeah. What's this person?
Well,
Because I don't see the chat when I'm doing.
Pau Puck.
Okay. Yeah, you should come on this show sometime
Like
because that's exactly right. It's just like it's kind
an
of like a
old
telescope.
fashioned telescope. Old fashioned.
Yeah.
Like those little things that the captain on the ship has.
Mm-hmm.
Yeah.
Yeah, and so you can do these detailed molecular analyses of exactly what genes are present in the fly wing, and you can map them all out, and you can see some of the underlying basis of the pattern. So fly wings are not as spectacular as butterfly wings. So we don't actually see those spots. But the spots that you see in a butterfly are formed by the expression of distal-less. But they're not going to form a leg or a wing, right? They're just going to form the scale pattern there. So it's a beautiful example of recycling genes. So we pull out a gene that is essential for forming large scale structures like a limb, and we re-express them in a different role in a different environment to do things like control color spots. So when you look at the fly wing, you see all these cool things like, again, these are really familiar genes. Engrailed is that's one of the segment polarity genes. So it's expressed in every segment in a particular region. And that defines the posterior compartment of the wing. And then there's hedgehog. We've all heard of hedgehog. Yeah, that's expressed out here. Then we've got these other genes. So here's distal-less. Distal-less is expressed at the intersection of wingless. Again, that's kind of a logical name, right? Wingless. If you have a mutant wingless, you don't make a wing. But it's expressed in this band here. And then here's this, I love this gene, decapentaplegic, which is another gene that's really important.
Let's deconstruct it. Deca is 10.
10.
Penta, and that's many. Plegic, without
Oh, it's more
legs.
specific than that. It's
Oh.
10 and penta for 5.
Oh, Penta. Oh, yeah, right.
So this is the 15 gene.
Oh, right.
How many imaginal discs are there in each side of a fly?
15.
Yep. So this is a gene that's expressed in all of the imaginal discs, and it's a gene that's important in defining the orientation, the polarity of that disc. So these genes here, decapentaplegic and wingless and engrailed and hedgehog, are basically defining the positions on the wing, whether you're at the base or at the tip, front or back, dorsal or ventral, all this kind of stuff is defined this way. And then we have decapentaplegic and wingless working within that system. I hate to use this word because it gets so misinterpreted by Jordan Peterson fans, but there's a hierarchical pattern of expression. So you turn on one gene to find a broad region, and then you start divvying it up by expressing other genes in different orientations.
I think the correct term is like a modularity.
Yes, so you've got a module that defines distal part of the wing, and you've got the intersection of modules that generates a new module, like this
So the
pattern of distal.
modularity can be summarized as to each according to its ability, to each according to its needs.
I think of modularity more as a combinatorial toolkit. So you've got all these different modules can be assembled in different ways, and that allows you to generate these novel patterns.
Anyway, no more political jokes. So just keep focusing on the science for now.
Yes. Okay. So anyway, that's the level of detail I just wanted to present today, and then I just want to briefly mention some other things besides butterflies. I mean, this is one of the things that's got me interested in spiders. is I'm looking at spiders and asking, hey, there's a pattern here that when I look at these animals, what I see is, here's these kind of circumferential patterns in the abdomen. Uh, Different species, you can also see that there's a dorsal midline stripe, but different species have different patterns of expression, but it's like there's a set of rules there. And when you actually look at these things develop, what you see is in the really early instars, this starts off as a dorsal midline stripe with little nodules of color stuck to it that then expand circumferentially. So that would be an interesting question to pursue. I think we're going to see the same sort of things we're seeing in butterfly wings. Another example that I want to bring up is Blaschko's lines.
Oh yeah, yeah. I was also thinking about that as well. With the
Yeah,
yeah.
Blaschko's lines are really interesting because human beings tend to be kind of bland in our coloration. Again, typical mammal, right? We've got some things like counter shading, like the underside of our palms tends to be lighter than the back of our hands. It's especially distinctive in more darkly pigmented people than us Minnesotans. But yeah, than us white Minnesotans. But yeah, that there are some general patterns, but there's also these subtle things that are only rarely exposed. So Blaschko's lines are appearing in this woman. She's got a pattern of skin nevy, little moles and warts and things that have this interesting swirling pattern all over the place. So like this V-shaped thing down over the abdomen,
Mm-hmm.
that's characteristic. So is some of it that you see here, but you also see some other complication things going on here. And what is thought to be going on here is that these represent lines of migration of epidermal cells during embryonic development. So
In what direction is it? Away from the center on the back,
yeah,
or
so a lot of these are going to
towards? Yeah.
be, yeah, so these are going to be largely neural crest derived pigment cells. So
Oh,
they
so
would,
so away from the back, like
right.
right.
So they would have arisen in early development here along the dorsal midline.
Yeah, and
And then
that's why you have the V shape on the other side, because that's where they come towards each other. The V shape,
exactly.
right?
So they're moving out laterally, and also the body is elongating. So you get these V-shaped patterns. In zebrafish, I've looked at this in the past where you can actually, because zebrafish are nice and transparent as embryos, you can just focus in with Nomarski optics and you can actually see the cells and watch them migrate out in this pattern from the dorsal midline around the body. Uh, but again, you don't necessarily say pigment expression along those lines. It's just that in some of these rare cases that some individuals, what it may be is that in this person's embryonic development, there was one of the early cells had a
Yeah,
mutation.
just
And
one
so,
cell. Yeah.
yeah, and then that contributed to a population. And so there you got this intermingled population of mutant and non-mutant cells that produce this pattern.
You have also, like, I don't know if it is related to this one, but you have also like the X chromosome phenomena, where you have like in during embryogenesis, like when you have like 100
Yes.
cells or so, then each cell turns one X chromosome off and the other one is activated. Of course, if you could see the difference between which one is turned off and on, you would see stripes in women.
Mm-hmm.
But of course, there is no pigment linked to the X
Yeah.
chromosome in humans. Then you don't see any pattern normally.
Right.
But
Although there
you can
is,
see them. You can see them in calico cats. That
yes,
yes.
cats do have that X-linked pigment gene.
Mm.
And you can see it in some humans. There, what was the name of it? There's,
Sweating, I think, right? Like you
yeah,
see, yeah.
there's a disease that, a genetic disease that affects sweat gland formation, which isn't sexy at all, but you can see the patterns emerging on people's skin with this, particularly in women, uh, where it's patterns of dry skin against normal skin.
Mm.
So that's not so great. Uh, I think it would be spectacular if humans had the kinds of variation that allowed us to have, more than just solid colors, but
Yeah.
all kinds of interesting. Yeah, wouldn't it be cool to be
Aren't you? Are you saying that we are not special?
we lack these special traits. Yeah, but I would love to be leopard spotted. That would be awesome. But it's just not in our genetic recipe, it seems,
Yeah.
unfortunately.
So it's also important to note that even though we don't like necessarily always like visibly express these patterning, we still have the underlying mechanisms of these patterning. So
Exactly.
it's like the components are already there. It's just like a few tweaks, and then you have a pattern
Yes,
basically. So patterns can rather easily come from these processes.
that's the whole lesson of the butterfly is
Yeah.
we don't think the underlying pattern evolved to generate the colors. The underlying pattern was there, and then subsequent changes allowed for expression of that pattern. So that's another cool feature. Again, modularity, right?
Yes.
Yeah. Another great example.
Acceptation. Lots and lots of acceptation.
Yes. So here's another example I like to throw out at my classes: male pattern baldness.
Mm-hmm.
There is so much variation in this. Different people have different patterns of baldness. I mean, there's some that are more common than others, but there's just, you can have the receding forehead. Uh, what runs in my family is we get this increasingly prominent widow's peak. It all recedes around here and it curls around the top of the head, leaving a little knot of hair right here. I have fortunately avoided that. But yeah, you get all these different patterns. And what those tell you is, again, I don't think this is something that's been selected for. This is not something
No.
that has an adaptive significance.
This is
But
an
what it
example of unfortunate neutral evolution. At least
Yes.
unfortunate for men. For some
Well,
men.
but it tells us that there's an underlying pattern of sensitive, for instance, testosterone affects this. So there's an underlying pattern of testosterone sensitivity that varies in an interesting morphological way. We just don't exploit it in any way. We don't use it in any way.
Yeah.
But for instance, this is obvious that you go in, you poke here, and these cells are expressing hair far more than cells up here at my cheekbone.
Mm-hmm.
Why? And it varies in different apes, of course. So there's all this interesting variation going on. And then of course it also varies with sex. That what we can suspect is that women out there also have this variation in receptivity to signals. And we know this because women who take testosterone don't suddenly grow hair right here. They grow it in the typical places for male hair growth. So there's again there's this invisible pattern on our faces and our heads and the rest of our body that isn't utilized to generate any kind of interesting visible patterns, but it has the potential in the future. So, again, not that I think it's necessarily going to happen, but just there's all
So,
this cool.
some people in the live that are lamenting where they belong in this chart. Some say, "Oh, I'm VA," or "I 4A."
You don't lament it. It means that you are expressing your
I'm
your patterns more strongly.
I'm lucky so far. I'm belonging in like one, the first one. So
Yeah, and I mean
yeah.
I've got some thinning up here, but not a lot. And I know my great grandfather, for instance, had a full head of hair until he died in his late 90s, and it kind of looks like I'm falling into that pattern, which is good. But there's nothing wrong with these patterns. These are I wish we could somehow as a society, celebrate this variation within humans, and there is nothing wrong with any of these patterns. It's just, to me, it's fascinating. And I see a balding person and I want to just sit down with them and explore their scalp, which is kind of creepy. I know I
Yeah.
wouldn't do that, but okay. So let me get out of this and uh-oh. There we go. There we go. Now I'm back to normal, and now I can read everybody's comments again. That's one drawback to using the Zoom and Keynote, is I'd lose the chat. Oh yeah, I see people trying to characterize their patterns. I love you as you are. You're all beautiful. Doesn't matter what pattern you've got. Oh yeah, there's the idea that balding represents old age, right? That so does graying. So I'm
Yeah.
familiar with this.
Also, it's an interesting pattern to see with graying. Like when people gray, the eyebrows usually stay black, and also the beard. Maybe some parts of the beard stay black.
Yes.
Yeah.
Yeah, but then even within that, there are differences in patterning. So, for instance, my eyebrows tended to go gray pretty early, which is unfortunate. Uh, I got a little hint of it in my temples that started showing up when I was in my 20s. And when the beard went, it went in stripes. So, oh, Nestle has some illustrations for us too. How the mouse makes waves. Oh. Those aren't mice at the top. That's a dog and a raccoon and Sean Connery. But okay, yeah.
Yeah. So this is all really interesting pattern to note from what you can see in mammals, even us.
Yes.
Yeah.
Yeah. So that's what's interesting about this particular topic. Pigment patterns are superficial, but they're relatively easy to access, which makes them really appealing. And then in many cases, as the Protas and Patel paper points out, these often have adaptive significance, right? So
Yeah.
you kind of need these for things like camouflage and Batesian mimicry, and um, there's really important in things like mating displays.
Yeah. Also,
So
there are some like controversy. For example, we spoke about the zebra last time. And of
uh-huh.
course, some people think, oh, they might be a form of camouflage, or maybe like if a lion sees a herd of zebra, then the pattern will make it look like they are all one creature or something. Like I don't buy that necessarily.
Right.
There's also like a hypothesis about like the fly, like flies that infect animals, and then when they see these patterning, they get confused or something. So it's really a mystery about why zebras have these stripes.
Yeah, I mean, I've read some stuff about the significance of disruptive coloration, which I find credible, because as a typical vertebrate, when I'm out looking for things, and I bet the audience out here is all familiar with this too, that you cultivate search patterns, that when you're out looking for things for specific things, your brain begins to adapt and recognize them. So when I was out fossil hunting, for instance, I started to pick up on what a fossil would look like, color and shape and size and all this sorts of thing. And so you start to cue in on that. And one minute you're walking along, you're wondering, where are the fossils? And then the next minute you're saying, oh my gosh, they're everywhere. I'm surrounded with old bones. And it's the same here with a zebra, that the idea is that the predators have a generic search pattern when they're looking for tasty mammals. And if you've got a tasty mammal that's got all these stripes on it, it disrupts that search pattern so that it doesn't jump out at you that, oh yeah, there's a four-legged mammal with these wonderful fat haunches I could bite into. Nope, all you see is the patterns of light and darkness. Okay, do any
More
of our
questions in the live chat? Oh, someone says a predator prefers you aren't looking at them, I guess. Oh, that,
yeah.
yeah. Oh, so what about the color changes in chameleon and cuttlefish?
Oh, right.
Like active patterning, like in real time,
Yes.
basically. Yeah.
The,
Although not maybe not exactly the same. Like of course the structures still stay in place in the skin, like for example, but still there is of course some pattern control happening in there. Yeah.
yes. Yeah, that again, there was a really good study I read, and I can't remember the author now, on the camouflage in cuttlefish. And there it's a beautiful example of disruptive camouflage, because it turns out they are not trying to directly emulate the surface they're on. What they're trying to do is sort of develop a pattern that's roughly modeled to the same degree. So they tend to blend in that way. So yeah, there's that there you can imagine the way that evolved is by first acquiring control of the degree of pigmentation. So you can imagine an ancestral cuttlefish that just would model itself, either light or dark, depending on the environment. And then having individual control, you can start thinking about, well, yeah, now I can make it look like I'm covered with pebbles or covered with stripes, whatever best matches the environment.
Mm-hmm.
Yeah. So that's how that is. What that really represents is the evolution of exquisite neuronal control of pigment cells.
Yeah, some are also mentioning the hypotheses about the zebra stripes, like the one like the flies, making it harder for them to, and also the herd here, like a striped zebra is invisible among all the striped zebra.
Yes.
Yeah.
Yeah, the tsetse fly hypothesis, I I read one paper on it a while back, and I wasn't really persuaded, in part because, and this gets on into our discussion of, what makes for good science and all that, is they've got some empirical evidence, some statistical evidence of differences, but we don't have a mechanism.
Yeah.
What is the mechanism that
Also, are zebras like really plagued by these flies? Like, are they really infected by really severe diseases from these
yes,
flies? I don't know. Like, yeah, maybe it can be plausible explanation,
right,
but yeah,
yeah. Oh, I see another question here. Are there color patterns that fall outside the typical human visible light spectrum, such that they don't appear to have patterns to us, but are discernible to other
yes,
organisms? Yes. Yeah. I'm particularly an example I'm thinking of are flower petals.
yes, exactly.
Yeah, that they're
This is more close to my area. Yes, like we see, of course, a lot of different patterning in flowers for obvious reasons, and there are also places in the flower where it differentially reflects UV light, so it also has like a pattern in the UV spectrum also.
yes. Yeah, and also some of them vary the plane of polarized light. So we can't sense plane polarization of light, but there are animals that can. So
Yeah.
lots of tricks we can play.
Yeah. It's also maybe important to point out, like, again, I have like an old gripe against the concept that like the genome is like a perfect, uh, like a blueprint of the organism. Like you can have a one-to-one correlation between a gene and a trait, and the pattern formation is like one of those things that shows no, it is not that simple at all. Like
Yes,
you cannot see the patterning instructions in the genome at all. Like
correct.
try to find the pattern instructions. You cannot find them.
Yeah, so butterfly wings, for instance, what you've got to do is generate this flat sheet, biologically. And so what you could easily picture is that there are molecules that are diffusing out to generate positional information on distance, but they ripple and they interact with genes that are defining anterior posterior, for instance. And so you get these complex patterns out there that are not encoded for in the genome. They are a consequence of molecular interactions during the formation of the structure.
Yeah. So again, it's an emergent process, and also there's a lot of like structuralist processes are going on, of course.
Yes.
Like of course, but bear in mind, structuralism, like to an extent, is maybe too far, of course. Like again, the idea that like structuralist guides evolution, maybe too far.
Yes.
Of course, it may stay like puts limits on what is possible. Maybe, yeah, of course. Structuralist
Uh-huh.
principles, yeah,
Okay, you have touched on two magic words for me, that emergent properties and structuralism.
yeah,
We don't want to get into that today. Maybe some other time we can have a discussion about
yeah.
structuralism.
Of course, like pattern formation, like some pattern formation involves like the activator inhibitor interaction,
Yes.
generating the Turing pattern, for example. Yeah,
Uh-huh. Lots of things that we can
yeah,
explore in the future. So let me wind it up here. We've been going 45 minutes. I think that's a good length of time.
yeah. We don't need to
And
have like a 2 hour, two
no,
hour
I don't.
one. No.
I do not like 2-hour live streams. So 45-minute live stream is just about right. So let me just say a few things. First of all, I'm going to urge everyone to like and subscribe to the video. Yeah,
Mm-hmm.
do that. It's good. It'll help things grow. I noticed, you know, we had this little discussion about how we can improve traffic and things like that. And I noticed when I was reviewing my videos that my spider videos got more traffic than my Evo Devo videos.
Mm.
Yes. So I was thinking, I wouldn't mind if I just switched to doing nothing but spider videos. So if
Maybe video about the evolution of silk, spider silk,
yeah, I could,
or the development of spider silk.
I'm thinking just aim a camera at spiders and let them run. Uh,
Yeah.
so unless you want to avoid that fate,
Yeah.
I
Also,
mean,
there's one last question, like
oh,
scorpion light
oh,
under UV. Like, why do they do that? I have no idea why.
yeah, why? There isn't a why. I think many times in evolution, it's a mistake to ask that why question because then it leads you
Yeah.
into adaptive solutions.
Yeah.
I bet you that's
Of course, like a random variation is also a why.
yes,
That is valid.
yes,
Yeah.
yeah. That scorpions, like other arthropods, have to tan their cuticles. They have soft cuticles when they first molt, and then they secrete enzymes that cross-link things. And I bet you there's just some enzymatic activity that worked well enough to toughen the cuticle and also coincidentally
Yeah.
produced these
It may be also the reason why our blood is red. Like it's not red because it's like it's beneficial for our blood to be red, but it's just like we happen to be using iron as our oxygen carrier
yes, exactly.
cofactor, and then that makes our blood red coincidentally.
Okay.
Yeah.
So as I was saying, no, I'm not going to do just spider videos. But I just so you can help me out by liking and subscribing and all that kind of good stuff. Then next week, I have plans to discuss another paper by Nipam Patel. Yeah, Patel does really good work. I've known him for years, and he's one of these guys who just goes out and explores all these different organisms, and it's great for looking at diversity. He does a lot of Evo-Devo stuff. Anyway, I want to talk about a paper by Nipam Patel on evolution of the arthropod nervous system.
Ooh, there's also interesting fossils about that as well, actually.
Yes,
Interestingly enough, like you wouldn't expect there will be fossils about the nervous system, but there are.
right.
Yeah.
And there's also molecular fossils all over the place. I mean, uh, I worked on grasshoppers and I've worked on Drosophila, and it's astonishing when you look at the embryos, they have the same pattern of neuroblasts with subtle differences, and they just diversify as they get older. So you can actually see a primitive pattern, a consistent structural pattern that's present in all kinds of larval and embryonic invertebrates, just retained for 500 million years.
Hmm.
So that's the plan for next week. So tune in then. And yeah, Nestle, if you want to join in, that's fine.
I will see if I have the time.
Yes, I know it's a time constraint. I actually
Yeah.
do have to change the time that I'll be doing this.
All right.
Still planning on Wednesday. So I'm trying to decide whether I want to do it earlier or later, because this time next week I will be teaching genetics.
No way.
Yeah.
So
So which
this session for me it started at 7 p.m. So if
oh
you if you plan like a lecture like 5 hours later, then it will
no
be
no
difficult for me, of course. Like yeah,
I'm thinking, like after my class or before, I got to have like an hour buffer
right.
between this and the class. So I'm thinking like maybe 2 hours earlier or 2 or 3 hours later.
Hmm.
There are these. Come on, people! I'm supposed to be winding this down, and you keep bringing these things. Yeah, you can get resins that cure under UV light. I go to the dentist and look at what they're doing there. University of Oregon has learned how to create colors using molecular shapes. That, as a U of O grad, that's interesting. Is this work being done with molecular probes? That big chemical company out there?
Hmm.
Anyway, yeah, there's all kinds of interesting
Oh yeah,
things we could talk.
I'm just the shape like some butterflies have particular shapes in the scales that reflect blue light.
Right.
Right. Yeah.
Yes.
Of course, now we are opening a new discussion again. So
Oh
trying
no.
to winding now. Yeah.
Yeah. But yeah, it's like a lot of vertebrates do not have access to a variety of different colored pigments. And instead, what they do is they generate microchannels and scales and so
Yeah.
forth. For instance, they generate iridescence patterns and cool stuff like that. But for real, I'm going to stop right here. Keep them wanting more. So come back next week. I'll make an announcement about the specific time. But next Wednesday, we will be talking about development of arthropod nervous systems. And I will tell everyone as well that you don't have to just be in the chat. If you know something about the subject and you want to talk about it, send me an email and I'll send you a link so you can join in too. And with that, I'll say good afternoon. We'll talk to y'all later.
Now I will say good day, good evening.
Yes, and thank you so
Yeah.
much for stopping by and making this a little livelier.
Yeah.
Yeah. Okay, talk to y'all later. Bye. Okay, I think we're